Hello, welcome to this drop-in session. Are we live? Just give me a thumbs up if you can hear me okay. Perfect. Cool. So also quick show of hands. Who's been to one of these classes before? Maybe for another subject. Doesn't have to be biology. So who's done one before and who is here for the first time? Okay, so quite a few of you guys have done the classes before. Nice. Anna, it's your first time in Paris. Chloe, welcome. Welcome to Snaprovise. Oh, quite a few first timers actually. Nice, exciting. So basically, yeah, we're going to run through a web class this evening. We're going to spend 1 hour going through some biology. The focus today is on cell membranes, so it's a really important part of biology. It's in all syllabuses, all specifications. You're hearing me double. Is that for everyone else as well? Thanks for letting me know. Let me think what the issue with that could be. Oh, so it's normal for everyone else. Okay. Okay, that's good. Good to know because I actually wasn't sure how I was going to fix that problem. So that's good. I'm not sure. Sorry, who's having the problem? Maybe try plugging in some headphones. Sometimes that can fix audio problems. Elsie, yes, this will be recorded and it will be available to view after the session if you'd like. Yeah, as Fizna says, maybe also try like leaving the meeting and rejoining if there's any issues. But it's usually all right. Oh, you had 2 tabs open. Okay, that will explain it. Nice. Well deduced. So yeah, it's already like 2 minutes past. So I think, well, everyone who will be joining is probably already joined. So let's just dive straight in with our lesson for today. So I'll share the screen. So as mentioned, we are going to look at membranes. Hopefully you guys are all looking now at a PowerPoint and we will go through. So a little bit about me first. I'm Alex. I'm the head of biology here at Snaprovise. I've got a degree in biology and did a PGCE also in biology. And I've been teaching and tutoring for the last 4 or 5 years. What spec? We're doing it. This is for all specs. So it doesn't matter if you're doing OCR, EduCAS, even like international A levels, this will be relevant for everybody. But yes, it's pitch and A level. So it's like year 12 content technically. Okay, so also we're running a little competition today. So we're giving away an account for you guys. So very exciting. You've got a chance to win a free account. So to be in the running, all you have to do is follow the Snaprovise Instagram and then share a picture of the web class you're attending this web class, and then just tag Snaprovise, and you can be in a chance to win it. Funka, you already have an account. Well, that's great. Maybe you can still enter. Maybe you'll get a free. They'll like make your account free anyway. I'm not actually exactly sure, but there's no harm in entering. Or sign up a friend and use their free account or something. So does the free account have all subjects? That is a good question. I'm actually not sure whether it's for one subject or all subjects that you need. That would be something to ask if you win, I guess. And then, yeah, so we're giving away a free account, and we're also, this is for everybody, this one, we're giving a coupon code, which will have a discount. So everyone's going to get the coupon code, and then we're going to give that at the end. So stay tuned for that. Okay, so that's a little bit of admin. Let's dive straight into the lesson. So some of you, I recognize some of you from the classes. So some of you will be familiar with the format that we normally do for biology. And for those of you who aren't, I'll just explain what we do. So we normally start a session. So the first like 10, sometimes even 15 minutes, we do some refreshers and recap on the topics we've done previously. So this is from a series of lessons that we do weekly. And in the prior lessons to this, we'll have been looking at mitosis and meiosis and also on enzymes. So that's why you might be thinking these questions are a bit random. They don't really have anything to do with membranes. We just always do like a little recap to keep the content familiar and to refresh it. So it's really important to like embed these concepts. The syllabus for biology is huge. So the real challenge is just getting all that information into your brain. So this is a really good way to help with that. Will we do more free classes? We do once a week for each subject. So actually once a week for a subject. So for each subject, because there's 4 subjects, we do it once a month on YouTube. But on our actual website, so the subscription package, we do 4 classes a week per subject. So there's a lot more classes if you're actually like a member of Snap Provides. Do one on organelles? Yeah, we could do. I can't remember what the next one is actually planned to be. Oh, it's in the notes. It's like, yeah, there's a list of the schedule. Lavinia, yes, this is relevant for CIE. We don't have a specific CIE package, but the syllabus is very similar to the ones that we're working with. So yeah, it's all going to be relevant for that. Okay, anyway, I could waffle on for ages. So let's dive into some biology. So first question, and this is open to all of you guys. So the more you interact, the more you get out of it. What is the equation for calculating the mitotic index? What's the equation, and why would we calculate a mitotic index? What's the reason? D, all that stuff we'll talk about that later. Funko, you don't know. Okay. Chloe, yes, very good. Nice. So the number of cells in mitosis, undergoing mitosis, divided by the total number of cells. And Anna also got that there, and Alexia. Nice. So, hi, Rush. Oops, I've written the second part first, by accident. So the cells in meiosis, mitosis, divided by the total number of cells. And that will give you an index of how many cells are undergoing mitosis. And we can use that to determine how quickly a tissue is growing. So if it's growing fast, it's going to have a high mitotic index. If a lot of cells are undergoing mitosis, the tissue is obviously undergoing rapid cell division, so it's growing. Yes, this is on all specs, the mitotic index. So can you guys think of a medical use for this? Why would you want to know how quickly cells are replicating or how quickly a tissue was growing? What would be a medical benefit? Yeah, nice. So yeah, we can use it to look for cancerous tissue and look for tumors. So we take a sample from the tumor and we see how quickly it's dividing, and that'll tell us how severe the cancer is. Also, it will tell us if the cells are cancerous in the first place. Yeah. So the faster that cancerous tissue is dividing, the more of a worry that cancer is. If it's growing slowly, it's not such an urgent threat. It's not as likely to be life-threatening. So yeah, it's really important for that. And they do this in cancer wards. Is it oncology? Is that the term? So it's a key part of that. Okay, next question. Why are isotonic solutions important in biology? Homeostasis, yeah. The difference between tumor and cancer is a tumor is actually what we call the tissue that's made up of cancerous cells. So they grow in different areas of the body and they can spread. When they spread, that's called metastasis, and that's a concern. But yeah, so that's the actual like cancerous cells, the growth of them are called a tumor. And yeah, liking these answers coming in. So we use isotonic solutions to, well, yeah, regulate osmosis or prevent osmosis. What would we say? I'd say like maintaining osmotic balance is okay. So we kind of do this to prevent osmosis. Because if you don't have a concentration gradient and an isotonic solution, you don't have a concentration gradient. This will prevent osmosis from happening. So we prevent osmosis. If we were studying cells, say, like looking at animal cells, this would also prevent osmotic lysis. So you guys might have done a study with where you looked at blood cells under a microscope. You can look at your own blood cells. You just take a blood sample and look at them. What you'll notice, if you put blood cells into water, they're going to swell up and burst. So we call that osmotic lysis. So lysis is like bursting, and osmotic is because it's bursting because water is moving in. So if you put it in pure water, which has a high water potential, water is going to move into the blood cell and it's going to swell up and burst. So not very useful for looking at the cell. So we put it in an isotonic solution, which has the same water potential as if it was a blood cell. We look at the same water potential as blood, and it's going to stop the cell swelling up. Ethan, it's a good question. In plant cells, it's a little bit different. What happens in plant cells if you put them into pure water, and why do they have a different response? Yeah. So plasmolysis is actually the opposite. That's where it's the cytoplasm is pulling away from the cell wall. If you put it into pure water, something with a high water potential, it's going to become turgid, exactly, and it's going to push against the cell wall. But the membrane isn't going to burst because the cell wall is providing support and rigidity. So that's the difference between plant and animal cells. Bacterial cells, also the same. They have a cell wall, so they don't burst due to osmotic lysis. Okay, next question. So a little throwback to mitosis. It's a funny way of wording it. How can one identify anaphase in mitosis? How would you know that the cell is in anaphase? What would you say for this? Chloe, are they lined up in the center or? Yeah, they're going to be like moving towards the poles if they're in anaphase. So moving towards the poles or at poles, to or at poles. And what shape are the chromosomes going to be in? I think I saw Anna. Yeah, nice. So that they're in this very characteristic V shape, and a few more of you got the answer there. Yeah, so they're being tugged by spindle fibers, which are attached to the centromere. So that's the spindle fiber. I'm just going to put SF for short. It's not actually a real acronym, but we'll do that for now. That's the centromere. I leave myself much space to write, not very clear. And so if it's being pulled by the centromere, by the shortening of the spindle fiber, it's going to be forming a V shape. So that's what we look out for. Rather than the characteristic shape of a chromosome normally is more of a sort of like sausage or rod shape. I don't think sausage shape is a particularly scientific definition. Maybe we'll go for rod shape instead, slightly better. Okay, so hopefully that makes sense. And next question, on to enzymes now. So how does a competitive inhibitor affect an enzyme's rate of reaction? What do we think? Oh, some of you got it there already. Yeah. So it's going to slow the rate of reaction. So it's going to reduce it. But it's not going to completely stop it. Yeah, so it's going to decrease it, but still the reaction will still take place. Still occurs. And yeah, as you guys are saying, a competitive inhibitor has a similar shape to the substrate. Therefore, it's able to bind to the active site. So I actually quite like the enzymes topic. It's really simple and it's really easy to score marks on it because the mark schemes are like so keyword specific for this. So if you talk about complementary shape, active site, enzyme substrate complexes, if you basically just throw out those keywords for nearly all answers, you can score like pretty much full marks with these things. So yeah, just learn the vocabulary. How do enzyme substrate complexes form? So because of their complementary shape, the active site and the substrate bind. And we call this the induced fit model. This was like a revised theory on the lock and key hypothesis when we realized that actually proteins aren't rigid, they're more flexible. So we upgraded the model. And yeah, they bind. And as the enzyme sort of reforms its shape as it binds, that's what actually stresses the bonds in the substrate and either splits it apart or joins them together. It's either a catabolic or an anabolic reaction. So a couple more keywords there. I'm throwing a lot of keywords out. I just wanted to say, I saw somebody put, Ryan, just be careful. It's not complementary base pairing in enzymes. That's in DNA. So yeah, just be careful with that one. Thought I would point that out. Okay, so and onto this question. So why does a competitive inhibitor affect an enzyme's rate of reaction? Simran, yeah, I would talk about that one. Some of the specs, they like to ask about why the lock and key wasn't correct. So just be aware that it's because proteins aren't rigid in structure, they're flexible and they can move. So be aware of it, but they would normally talk about the induced fit model. Yeah, exactly. So the inhibitor is bound to the active site because it's got a similar shape to the substrate. Therefore, the substrate can't bind. So it's going to reduce the number of enzyme substrate complexes. Number of enzyme substrate complex. Or actually, I don't know, I've written that kind of weirdly. Quite often you see it just as like ESC. This actually is quite often accepted in the mark scheme, just writing ESC, but I would always recommend just writing it out fully, at least the first time. If you're doing a longer answer question, like an essay style one, you can write it out fully once and then put it in brackets and then refer to it as ESC afterwards. But yeah, better to write the whole thing. Ryan, yes, we have GCSE ones on Tuesdays at the moment. So at 5, I think it's 5:30 on Tuesdays. We're doing them for the next few weeks. So if you're studying GCSE, you can join in for those. Cool. I'm aware, because there's quite a few of you guys, like in a normal class on the website, we have, well, usually between like 5 and 10 or 12 people in a lesson. And I can answer all the questions with this many people. I probably can't answer all the questions. So I'll do my best to answer as many as possible, but don't feel like rejected if I'm missing your questions. It's just because I can't answer all of them within the time frame. Okay, so yeah, what do you guys know about membranes already? Any keywords you guys can tell me about? So this could be a membrane structure, or transport across membranes is another part of this topic. Partially permeable, phospholipid bilayer, like those. Cell signaling, yep, they're involved with that. Fluid mosaic model, very nice. Cool. Hydrophobic, hydrophilic, yeah. Channel proteins. Nice. Yeah, loads of good words there. Cool. So you guys clearly know at least the basics of the topic. So let's go through and see how much we can remember. So the objectives of this lesson are, we're going to describe the components of a membrane and look at the fluid mosaic model. And then we're going to look at transport across membranes. So this is split into passive transport and active transport. So one is using energy and one isn't. So yeah, we'll look at both of those later. We'll see how much we can get through in the session. Okay, so these are the specs. It's just basically, if you want to screenshot it so you've got a record of it, then please feel free as I flick through them. They all, for this topic at least, they're all actually very similar. Like you need to know the structure of membranes and you need to know how things transport across them. So yeah, it's basically the same for all exam boards. Here are the Edexcel ones. There are, of course, like slight differences, but the stuff we're doing today is relevant for everybody. Educast, any Educast people here out of interest? They're quite rare. Or any like international people. Emily, you're Educast, nice. Don't see many people. Yeah, usually AQA and OCR are the most common, and then there's a few like Edexcel B, the SNAB people, and then like not like usually like hardly any Educast.
I'm not sure.
Cool. So part one, membrane composition and the fluid mosaic model. So let's talk through some of these things. We've got, well, this is a section of membrane, and there's different things embedded in it. So this actually relates to why we call it the fluid mosaic model. Anybody want to hazard a guess? This is quite often like a 2-mark answer on an exam. Why do we call the structure of membranes the fluid mosaic model? Yeah, we'll try and get through all of it today if we can. Which exam board is the easiest? They all have like their own things which make them like their own difficulties. I would say that OCR definitely has the biggest syllabus, which makes it tricky. But then because they have to learn more, usually the grade boundaries are lower for OCR. So to get a good mark in any of the exam boards is difficult. There's not like a really soft option. But they should really, I don't know, it is weird that there's different exam boards. They should kind of just have the same exam board for everybody, because it's in other countries, that's the situation. So it's a bit weird in the UK that we have all these different ones rather than just like the same for everybody. Yeah, what you guys are saying is all good stuff. Yeah, so mosaic, because the proteins are dotted around within the phospholipids. So, and this one, it's they are labeled. The green bits are the phospholipids. So within the phospholipids, we have all these different proteins and they're dotted around, and it looks a little bit like a mosaic. That's the name. So if you don't know what a mosaic is, it's like a type of art where you use little dots of tiles, that's not the word, pieces of tile to make an image. So really popular in like Roman and Greek times. I guess, are they still popular now? Maybe less popular. But it looks like that. That's why we call it that. And it's fluid because they can all move around. So they're not like stuck in their place. They can sort of move freely around the membrane. So let's do some of the functions now. So who can tell me what the function of a glycoprotein and a glycolipid are? What do these two things do? And what are they also? Yeah, nice. So they're like usually recognition sites. So they're to do with recognition and cell signaling, which is like communicating between different cells. So the structure of them, both of them are made up of, the glyco part is a carbohydrate chain. So it's the same sort of word as glucose and glycogen. So it's a carbohydrate chain. In this one, it's attached directly to a phospholipid. So that's why it's a glycolipid. In this one, the carbohydrate chain, which is the bit in green, is attached to a protein. So that's why it's called a glycoprotein. But they're both sort of have the same function: cell recognition, cell signaling, yeah, binding sites for hormones, that sort of thing. The 2 points, why it's fluid mosaic. Mosaic because it's embedded with proteins. Fluid because they can all move around. So that's the 2 marks, 1 for each. Nice. So what have we got next? Let's talk about the proteins. So there's 2 main types of protein in the membrane. We can classify one as this type, so we'll call it that, and another as this type. So there are 2 different words for that, and we're going to see which ones you guys know. Annoyingly, this is where the exam boards do differ a little bit. Some prefer to use integral and peripheral, and some prefer to use intrinsic and extrinsic. So that's probably why some of you will know one and not the other one. So yeah, we call these ones that span the whole membrane. So they go the whole way across, therefore they're good for things like channel proteins. We call these integral or intrinsic. So you don't need to know both terms, you just need to know the one that you are familiar with. So whichever one you've been taught is the one you'll need to know for your exam board. You'll have to double check the specs. I can't remember exactly off the top of my head which specs are which, but just the one that you're familiar with is the one you should be using. So these span the whole way through the membrane. So I saw a lot of people were mentioning channel proteins, and that's what their main function is. So these are channel or carrier proteins. So because they span the whole membrane, they can form pores and allow things to move through them. So this is also an example of a channel protein. So there's another integral or intrinsic protein. This other one that's only in one side of the membrane, we call that a extrinsic or peripheral. Whoops. Peripheral protein. So what functions could peripheral proteins have? They're not going to be any good for channels because they don't go through. So what could they do? Dilly, yes, a G protein. That is actually one of the things they could be. So we don't talk about these in AS, but if you are a year 13 student, you might be familiar with G proteins, particularly OCR do this. I don't think it's actually in the AQA spec, but this is to do with secondary messaging. So if you're just year 12, you won't have come across this yet, don't worry about it. But other things they could be, Pashtan, nice, yes, they can be enzymes. So a lot of enzymes are embedded in a membrane and it keeps them all in the same place. So for example, not all of the enzymes, but a lot of the enzymes involved with respiration are all in the membranes in the mitochondria. So it keeps them all together, keeps them near each other and allows them to work efficiently. So it's a good way to store membranes, to store enzymes. Like for example, the ones in our digestive system, they're like maltase that will break down maltose into glucose that can be absorbed. They're actually in the wall of the villi. They're embedded into that wall. So any maltose that floats past is then broken down and absorbed. Cool. The final one, another thing we could say for this is they can be an anchor point for the cytoskeleton. So cytoskeleton is like the skeleton of the cell, and there's fibers that crisscross the whole cell. So they have anchor points in the membrane, and they're basically proteins that are embedded. So this could be the different, there's a few different types of fibers, but yeah, they can be anchored in proteins. Anchor points for cytoskeleton. Okay, and last but not least, we've got cholesterol, which is represented by the yellow here. What is the function of cholesterol? Yeah. Nice. So stability or rigidity, you could use either word, of the membrane. So they make it more structurally sound. Yeah. So they can actually, yeah, saying maintaining fluidity is a good answer. So they can when membranes get too hot, they get more flexible. They can actually prevent some of the membrane breaking apart. But also when it gets really cold and the membrane is at risk of stiffening up, they also help to space out the phospholipids so that the membrane doesn't get rigid. We don't want to have a membrane that's completely rigid because it needs to be flexible. So it sort of sits, it doesn't sit between the bilayers, but it sits between the tails of the phospholipids. So it's sort of in the space there, and it prevents them from stiffening up too much. So I guess, the final thing we haven't talked about yet are the phospholipids. So these are the major constituent of the membrane. So they look a little bit like two-legged squids. So they've got, what's the name of this molecule that forms the top part? Anybody remember? Yeah, it's a phosphate head. So there's a few different things we could talk here. So this is the head, and this is the tail part. The head is made up of a glycerol molecule with a phosphate group on it. So that little red dot is the phosphate group, and the bigger circle is glycerol. So glycerol with a phosphate, the tail is made up of fatty acids. So the structure is very similar to a triglyceride. Triglycerides look like this, but instead of having a third fatty acid, they lose this and they have a phosphate group instead. So that's the difference. This relatively small change is actually really significant because lipids are very hydrophobic. They don't interact well with water. But if you stick a phosphate group on, which has a charge, a phosphate ion has a positive charge, this will now mean that this end, the head end, is hydrophilic. It can now interact with water, which is unusual for a lipid. The rest of it is still hydrophobic. So one end is hydrophilic, one end is hydrophobic. So the head, hydrophilic. The tail is hydrophobic. So these words, they're from Greek, like phobia and philia, meaning like hating and loving. So they have one part which, interacts well with water and one part that doesn't. So this is actually why they are so happy in a bilayer structure in a membrane. So if you think about this being part of a cell, if this is the inside of the cell here, you've got cytoplasm. And if this is the outside of the cell, you've got other things like tissue fluid or blood, maybe. It's going to be like an aqueous environment on both sides. So water environment out here and water environment on the cytoplasm side as well, because that's majority made up of water. So both sides, there's an aqueous environment. So the phospholipids are happy. The heads, which like water, are facing out into the aqueous environments, and the tails, which don't like water, are nicely sheltered away in the inside, away from the water. And that's actually a really important part of what's holding the membrane in place. So these properties. An easy way to remember hydrophilic head, hydrophobic tail. Yeah, I actually can't think of a particular way to remember those. If there's anything that, making like mnemonics is a really good thing, and making, usually like the sillier you make something, the better you remember it. So I'm a big fan of like making up silly rhymes and things like that to help remember different bits in biology. I would also really recommend like some bits you just need to memorize, and it can be a bit confusing. all those topics, make a deck of flashcards, and just each time you get to something where you have to just learn a few keywords or a few ideas, write it on a flashcard, and just every few days have a flick through your flashcards and just double check that you that you know them. So like read it, write questions on it, cover it up, see if you can repeat it, and you'll embed them pretty quickly if you do that. Exactly, it's like the arachnophobia. It's the same root of the word. Cool. So kind of a lot of information on that page now. That's like pretty much everything we need to know for the structure. So how are you guys feeling with that? Nice. So you can give a number, or you can give the emoji, whichever you prefer. I kind of dig the emojis. Nice. Yeah, lots of ones, lots of smiley faces. So that's good. A few twos, but hopefully we'll push you guys up to a 1 by the end of the class. Fatima, yeah, if you haven't heard of extrinsic, then don't worry. As long as you know the word peripheral, then that's fine. You don't need to know both terms. Oh yeah, I'm loving the dab emoji. I haven't seen that before. I wouldn't say cholesterol maintains an equilibrium. I would say it regulates fluidity, or maintains fluidity, or stabilizes the membrane. Either one of those two is the best term to use. Is glycerol a backbone? Kind of. It's not really a backbone. The structure of glycerol looks like this. So it's got 3 OH groups, and the rest are hydrogens. You don't need to be able to draw a glycerol out from scratch, but it kind of helps to visualize what's going on. So this is glycerol. Each of these OH groups can form a condensation reaction with a fatty acid or a phosphate group. So a fatty acid, if you remember, is a long chain of hydrocarbons, and then it's got a carbon with an oxygen and an OH. I've kind of drawn it the wrong way around because I was going to draw where the condensation reaction would form. So these basically combine with glycerol to form a ester bond. So we leave one of the oxygens there, we remove H2O, and the fatty acid and the glycerol are now joined, and we produce H2O. So it's a condensation reaction. And yeah, we learn about that when we do this sort of biological molecules topic at the start. Yeah, exactly, an oxygen bridge. Nice. So that happens 3 times to form a triglyceride, or it happens twice to form a phospholipid, and once with a phosphate group making a phospholipid. So it's these 3 OH groups that allow glycerol to form these 3 bonds. So it's quite a simple molecule, but really, really useful. What's an oxygen bridge? It's basically just the oxygen that gets left behind between, this is the carbon on the fatty acid, and this is the carbon on the glycerol. And this is the oxygen bridge. We can also call this an ester bond. Specifically for this one. Okay. So let's do some exam questions now. So the way we structure these lessons normally is a little bit of learning, going through a topic, and then we do some questions to just check that you guys are understanding. So in this one, scientists investigated the percentages of different types of lipid in the plasma membranes from different types of cells. They then show the results in the table. So we've got 3 columns here, well, 4 columns overall, 3 of the different cell types, and we're looking at the percentage of lipid in the plasma membrane by mass. These are the different lipids, and these are the different cells. So red blood cell, cells lining the ileum. What is the ileum? Where do we find that? And then we've got bacterial, like a bacterial cell. Yeah, it's part of the small intestine, involved, I'm going to put SI, involved with absorption. So that's where the ileum is, just in case you're wondering. So first question is, scientists express their results as percentage of lipid in plasma membrane by mass. How would they calculate that value for 2 marks? Will, yes, fire away with your question. You can use either Alex or Mr. B, whichever you prefer. But yeah, Alex is fine. Yeah, exactly. So I'm sure you guys are all familiar with this calculation. Well, probably the time you use it the most is when you calculate your percentage grade on a test. At least that's what I remember, like doing it the most at school. So to calculate the percentage that something is of the overall thing, so say you got like 7 out of 20 on your test. To calculate your percentage, you just do the marks you got, 7, divided by 20, and times that all by 100. And that will give you your grade for that test, which is not a particularly good grade. Maybe we should make it 17. So that's how you do the percentage on a test, and it's exactly the same thing here. So you take the percentage of that particular lipid. No, sorry, not percentage, amount. A particular lipid. And then you divide that by total amount of lipids. Oops, let's jump forward. So that's this part, and then you times it by 100 to get into percentage, because at first you're going to get a decimal. You times by 100 to get a percentage. So we can just do a really easy worked example. So we've got like 5, and overall we have 10 things. So 5 divided by 10, and then we times by 100, and this would equal 50%. I picked very easy numbers, but obviously, because I don't have a calculator to hand, so I wanted something I could do in my head, no sweat. But obviously you can do it for any numbers. Okay, hopefully that makes sense. Let's look at the next question. So it says, cholesterol increases the stability of a plasma membrane. Cholesterol does this by making membranes less flexible. Suggest one advantage of the different percentages of cholesterol in red blood cells compared with cells in the ileum. So hopefully you've got that. Let's look back. So we're looking at these two. Percentage of cholesterol is here. We're looking at these two figures, basically. So we can see that the red blood cell has more cholesterol than the cell in the ileum, the cell in the small intestine. Why do we think that might be? It's a little bit of a tricky one to think of. So I'd be interested to see what you guys come up with. Yeah, so yeah, some of you guys are getting you're on the right tracks. So they're free in the blood. What do you mean specifically by that? Yeah, nice. Okay. Asthma, yeah, you've added like more of an explanation now. So yeah, it's to do with that. So compared to the cells in the ileum, the red blood cells are like free floating, they're individual. So they're not in a tissue. They need to be stronger and have a more stable structure because they need to support themselves. If a cell is in a tissue, like in the ilium, it's being supported on all sides by the other cells. So the cells in the ilium are all supported. This is like lots of cells all together. They're supported by the other cells on the side of them. So they don't have to be as strong compared to a red blood cell, which is just floating by itself. That's a side view. That would be like a front view. Because they just float around in the blood, they're not supported by other cells. They need to have a more rigid structure, a more structurally sound. So hopefully that makes sense. The ilium is a part of the small intestine, Ryan, where absorption happens. So during digestion, obviously you need to break down the food. That happens in the stomach and the first part of the small intestine. And then the second part of the small intestine, the ilium, is where absorption takes place. So once you've digested it, you need to absorb all the nutrients that you've broken down. So you break down proteins into amino acids, lipids into triglycerides, and monoglycerides, and then you absorb all those materials in the ilium. So you might be familiar with like the villi and other features of that. But anyway, we're not going into that topic too much today. Yeah. So to get the answer, it's only a 1 mark question. So you just have to say red blood cells are by themselves and don't have the support of other tissues. Oh yeah, sorry, I didn't finish writing this. Dendritic cells and don't have of other cells. So something like that, doesn't have to be word for word like that, but something like that would get you the mark. So hopefully that makes sense. It's a bit of a weird out of the box question. Yeah. So because they need to support themselves, because they're not supported by other cells, because they're not in a tissue, that's why they do it. Melissa, it's a good question. Generally, unless it specifically says you're going to get graded on the quality of written communication, you can just put in bullet points. So for like, I think it's a really good way to do it. I always recommend doing like bullet pointed answers unless it specifically says you're going to be graded on the written communication, because it makes it really easy for the examiner to see where to award the marks. So I've done for a few years, like the examining for a few of the different exam boards. And yeah, as an examiner, I much prefer it when people do bullet point answers. It makes it really easy for me to award the marks, see exactly how they were trying to structure the question to score those marks. So I think it's a good thing to do. Okay, so this would be it for all specs again. That was, to be fair, that was quite a hard question. It was only worth 1 mark, but it was actually quite a tricky one. Okay, next question. E. coli has no cholesterol in its cell surface membrane, but despite this, it still maintains a constant shape. 2 marks for explaining why that is the case. So this requires some knowledge of bacterial cells or prokaryotic cells. Yeah, nice. You guys are all saying the same and correct thing. So because they have a cell wall, so that's 1 mark. And then for the second mark, you explain what the cell wall is made of. So confusingly, there's 2 words for this thing. So they have a cell wall made of murine, but you might know this substance as peptidoglycan. It's the same thing. So again, just you don't need to know both words, just know the one that you've been taught. So annoyingly, is another area where the specs differ a little bit. Some call it murine, some call it peptidoglycan. So just be aware that that could be a possibility, but you don't need to know both terms. So that's your second mark, and that's how we get 2 marks for that. Okay. How do you know that E. coli is a bacterium? Does it say? Yeah, here. So you might have heard of E. coli. It's a common bacteria that lives in our digestive system. But even if you hadn't heard of it, it did actually say it in the question. So you didn't need to know that. Piper, yeah, chitin or chitin. There's like 2 camps of people who pronounce it in different ways. I kind of use both because I don't really mind. That's in fungal cell walls, exactly as Pooja just mentioned. So it's another thing you need to know. What's the cell wall of a plant cell wall made of? You wouldn't be expected to be, you don't have to be able to name any bacteria. Oh, actually, no, that's not true. If you do OCR, you need to know the bacteria in the nitrogen cycle. Cellulose, yeah. Yeah, I think everybody needs to know the type of bacteria called rhizobium, which are the mutualistic bacteria that live in the root nodules and provide nitrogen for the plant. And then if you do OCR, you also need to know the other bacteria, unfortunately, like Azotobacter and Nitrosomonas. So yeah, unlucky for you guys. Luckily for everyone else, you don't need to know all those bacteria. It makes it slightly easier. Nice. So let's do how many time. Let's make a start on this. And yeah, we'll probably finish on this page. So the other like really important part of this topic is looking at how things transport across membranes. So let's just recap what things can move through a membrane and what things can't move through a membrane. Who can talk me through? Nitrogen cycle is a year 13 topic. So yeah, if you're year 12, you won't have studied the nitrogen cycle yet. Nice. Yeah. So small non-polar molecules. So e.g. O2 plus CO2 and certain hormones. So something like estrogen is a small non-polar molecule and it can move directly through the membrane. Yeah. Glucose, yeah, not particularly well. Water, again, not particularly well. Water can move through slowly, but doesn't move through very quickly. So if you want to move through water at a faster rate, you use channel proteins called aquaporins, which is another thing you need to know. We're not going to cover them too much now. So things that can't move through are ions, large molecules, anything with a charge, basically. So to move through, they need to move through special proteins called channel or carrier proteins. That's how they get through. Exactly. So that's what those intrinsic or integral proteins we looked at the start, that's what they do. They allow these like large non-polar molecules through. Okay. Oh yeah, as Franco mentioned, you can also use or endo or exocytosis to move things across through a membrane. So that's on like a mass transport basis. Okay, let's define these terms then. So we've got simple diffusion, we've got facilitated diffusion, and we've got osmosis. What are they? They are terms that you guys all need to know. And you just need to be able to like define them. Should be like roll off the tongue because you know it so well. Nice. So yeah. So we talk about the net movement. From high to low conc, something like that. So you can either say down a concentration gradient or from a high concentration to a lower concentration. Something like that would get you the marks. And the net movement is important because it's not to mean that none of the particles are going the other way, but in general, more of them are moving in that direction than the other. So they're always going to go down that concentration gradient. So these are all examples of passive transport. They don't require energy, but it means they can only go down the concentration gradient, which is different to active transport, which can go against concentration gradient. But basically, if you want to go against a concentration gradient, you need to put in energy. So you need to use ATP. Yeah, so facilitated diffusion is basically the same as this, but passing through a channel or carrier protein. So just the same as that. A transport protein, e.g. Carrier or channel protein. Nice. So that needs a membrane. Simple diffusion will happen anywhere. You've all seen simple diffusion happen. If you like spray air freshener in one corner of the room, and then you can eventually smell it throughout the whole room, that's simple diffusion. If you make a Ribena, like squash, it's gonna, like, the juice will all mix in. That's just simple diffusion. Facilitated diffusion needs to occur across the membrane, and it needs transport proteins to do it. So an example of that would be like absorbing glucose in the ileum, yeah, basically any of the substances you absorb in the ileum would be examples of facilitated diffusion. So the final one, osmosis, is net movement of water molecules from high to low water potential. So you can say water potential, you can say water concentration, either is fine. You need to be familiar with this symbol. We use this trident. It means it's the Greek letter psi. We use that to represent water potential. So be familiar with this one. So you can write it like that as well. Across a partially membrane. Okay, nice. Yeah, it's really important to add this last bit. If you're asked to define osmosis and you don't include that last bit, you actually won't get full marks for that definition. So make sure you add the membrane. You need to have a membrane for osmosis to occur across. It doesn't just occur in like a bucket of water. It needs to be across a partially permeable membrane to be osmosis. Okay, so there is a bit more. We're like, if we had more time, we would go through some of these graphs on the different types of passive transport and how they look different, and also some more questions. And we look at active transport. We haven't got time to do it today, unfortunately. Usually the pace is a little bit slower on these YouTube classes compared to when we do it with smaller groups. But yeah, and there's a few more questions, but I think you can download the packs. If you want to work through these on your own, you can also do that. So yeah, just going to walk you guys through the website and how it looks. So let me just close this one and tell you a little bit about, yeah, what what we're looking at. Sorry, I can't talk and think, so I have to do one or the other. Okay, so yeah, that did fly by, I would say. So this is our website. This is like kind of why we were running the class tonight. It was to show you an example of what we offer on our website. So if you enjoyed the session today, and I hope you did, and you'd like to do more of them, then we run these classes 4 times a week on our website. So it is subscription based. You need to have a subscription to be part of it. But I just want to talk you through some of the features we have, because you might not be aware of exactly what it looks like. So, oops, I don't want to add courses. I just want to look at my courses. That's the one. So let's look, let's take AQA biology. This is what the sort of basis of the website looks like. So the whole topic is split up, or it's split up by years, and then the practical stuff you need to know. It's all done into sections. So it starts at the beginning with the biological molecules, moves through those, DNA, ATP, and then onto the cells topic. And then I'm going to scroll to where we got to today. So we were looking at transport across cells in the lesson today. So let's have a look at this topic, since that's what we just did. So this content is, it's split up into topics, and what we have, which is a really useful thing, is before you start looking over the topic, you can do a little diagnostic quiz, and this will tell you how well you know the topic before. That's really important because, like I mentioned before, the spec is so big that you need to be able to target your revision where it's most needed. So this will tell you which bits you know and which bits you don't know. So you can target your revision to the most useful bits. Are year 12 exams cancelled? Yes, they are. They are cancelled for, oh, sorry, year 12 exams. No, if you guys are in year 12 now, you will have to do the exam. Well, most likely, no one knows for sure, but almost certainly you're going to have to do the exams next in like 2 years' time. But yeah, if you are current year 13s, they are cancelled at the moment. But it wouldn't be impossible that the government changes their mind. They certainly have done that before. So I wouldn't like rule out for definite that you're not going to have to sit some sort of an exam. Anyway, let's go through. Let's do this. So cell membranes are composed of phospholipids. So we click this. What type of barrier is in the cell membrane? So let's get one wrong for example. So do this one. It's gonna tell us later that we need to revise this. So what are we looking at here? Which cellular structure is represented in the diagram? Oh, sorry. And then which is a transmembrane protein? We would say, I'd say there's 2 in this one. Let's go for B. So, oh, there's quite a few more questions. I thought it was only 4. I'm just going to skip some of these. Skip. Obviously, you would do these if you were doing it by yourself. Pass. So this tells me that I knew these spec points quite well, and I didn't know these spec points as well. So this will tell me where I need to revise. So I can click on one of these, and it will lead me to a revision video directly on that topic. So it's also highlighted yellow, which is telling me that on the quiz, I didn't know the answer to these questions. So it's one I need to revise. Once I've watched it, and once I've answered questions on it correctly, this will go green. And it's a way of tracking your progress. It's all tracked for you on the website. So really, really simple. So you'll watch a few of these videos, do another quiz. If you then get it right and you can do some questions, it will go green and you'll be aware. So another part of the course are these web classes. So this is one side. We also run web classes, kind of like the class we've done this evening. So for example, we've got some this week. I'm doing one on Tuesday this week on mutation and meiosis. And so we do basically 4 sessions a week for each subject. So if you enjoyed this class and you found it useful, yeah, consider signing up for a package, and you can join in with as many of these classes as you like. Look at some of these questions. What do you get in the subscription? So you get a lot more with your subscription. We only do a very limited number of YouTube classes. So that's what you get extra, and you get access to all the quizzes and other content on the website. So yeah, you do get a lot more for that. We've also, there are some different packages. I'm just trying to find out where it says the prices. Somewhere. Is it on here? No. Basically, there's a few different types of package. So the most basic one just has the quizzes, the diagnostic quizzes, and the questions. The more advanced ones, the more so I think one's called, the top package is called the ultimate package. That's the one that has the web classes with it. If you get the one below that, you can ask questions and those get answered for you. So say you watched a video, you did a quiz, and you're not sure on why something is, or you've got a question on it, you can ask the question on the video. You can put a comment underneath it, and one of our team will answer that question for you. So let me show you what that would look like. So let's go back to our course. Let's look at a topic. I'm going to skip this quiz. So let's say we're watching this video. Catherine watched this video and she still wasn't sure on some, like one of the parts of the video. So she asked this question, and then one of our tutors replied with an answer. So if you pay for the questioning package, you can ask as many questions as you like. Oh, here's one that I answered. So yeah, hopefully that makes sense. That's the kind of different packages. Some of you guys are saying, why isn't it cheaper? Yeah, it is. Yeah, so it is like reasonably expensive. However, for what you're getting, I think it does represent excellent value. So compared to something like private tuition, which is, yeah, upwards from £50 an hour, certainly in central London, for one lesson. For the same price as that, for the ultimate package, you can ask as many questions as you like. You can attend more than 16 web classes in person a week. So 16 hours of lessons, all for the same price as like 1 hour of private tuition, not to mention the diagnostic quizzes and all that. So it's not cheap, but it is excellent value for what you're getting from it. And it will definitely, you'll notice an improvement in your grades if you engage with it. Yeah, so I hope you guys consider it, or you can give it a go. There's a 1 week free trial. So there's no harm in joining for 1 week and seeing how you like it and seeing if you want to carry on with it. So that was a little tour. Obviously, you can look at the website and yeah, do go through and have a look on your own. And the moment we've all been waiting for, the link to our discount code. Sorry, I just need to do this so I can see your answers. Yeah, you pay easily more than £100 a month for private tuition, so it is cheaper. So a couple of things. We've got more web classes. We do it once a month per subject, but if you do chemistry, physics, or maths, you can join in those other classes. Like to see that. Glad you're going to sign up. So set a reminder, subscribe to our YouTube channel, so you don't forget these. So yeah, just subscribe to Snap Revise on YouTube, set reminders. Here is your code. If you want £10 off your first month, you can use this code. So MEM10. It's only valid until midnight tonight. So go to our website, sign up. It's a free 7-day trial anyway. So give us a go, try it out, see if you like it. And you can also get £10 off your first month if you would like. Yeah. What's the next web class? I can't remember what we're doing next. It's not for a few weeks for biology, but yeah, it says on YouTube, on our YouTube channel, the upcoming classes, so you can check out on there. Cool. That's kind of everything I wanted to go through. So thank you guys for joining tonight. I hope you found it useful, and I hope you consider signing up to our website, or at least check it out, try the free trial. And yeah, I'll stick around for a few minutes. If you've got any questions, fire away. Otherwise, have a good evening, and hope to see you guys on the package on the website in the future, or in one of these sessions in the future. No worries. You're very welcome. Yeah, have a good rest of your weekend, or what's left of it, and see you guys in the future. Do you have any tips for homeschooled student teaching? Oh yeah, so this would be a great package for a homeschool student. It kind of the classes, you have the same people in the classes, you see them again and again, or not see them, but you sort of interact with them. So it sort of simulates some of the parts of being in a classroom environment, and there's loads of good stuff you can use. So I would say for a homeschool student, it's like even better than for someone going to like regular schooling. Can you use a discount code if you already have an account? I actually don't know the answer to that question, I'm afraid, Camden. You'd have to give it a go. I mean, I would always try using a promo code. Alan, that's you buy it per package. So yeah, they're going to be per package. Yeah, if you've got any questions, send them to this email address, hello@snapprovise.co.uk, and they'll be able to answer the questions related to like the specific sort of questions on the package. Yeah, it's not something I know all the answers to on that, I'm afraid. Okay, Glory. These live streams are always free for everybody, so you can always watch these. But if you want to do the ones that we have 4 sessions a week normally for each subject, if you want to come to those, that's when you need to get the subscription. So yeah. Jasmine, the difference between a channel and a carrier protein is that a channel protein is like always, well, not necessarily always open, but it's just an opening that molecules move through. Whereas a carrier protein, the protein undergoes a conformational change of shape to move the substance through. So that's the difference. A channel is just like a pore. Carrier protein needs to change shape in order to transport the molecule across. Ramin, yeah, they're £50, but I think if you subscribe to multiple, then the price goes down. So if you did 4 packages, it's not £200 a month. It drops down quite a lot. So there's a big discount for doing multiple. Azim, I'm afraid we don't have time to go into colonial expansion and selection today, but yeah, by all means, if you join in our other classes, we can go over that. We've got these sessions called drop-in sessions where you can ask questions. Okay, so yeah, I think we'll end there. Thanks again for joining. Please join the sessions in the future, and hopefully see you guys on the website at some point. Cool. Thanks again. Bye, guys.